All right, after last time, we're going to move on, and we're going to talk about the first stage of cellular respiration today. So after last time on Monday, we just kind of looked at a broad overview and talked about how we harvest energy from the breakdown from catabolic reactions, and we looked at oxidation reduction in biology, and we talked about how taking energy away from a substance by breaking them down, by breaking bonds and harvesting electrons. And we looked at how we're going to really look at the movement of H, of hydrogen, as the movement of electrons, since each hydrogen atom has one electron associated with it. So today we are going to begin our journey with breaking down glucose and turning it into ATP with glycolysis, the first stage of cellular respiration. So just remember what the whole point of this is. The whole point is to make ATP energy, all right? To harvest as much energy from the bonds that exist between the different atoms in glucose and to turn it into ATP, which the cell can use for energy. So if this is kind of the overall picture of cellular respiration that I shared with you last time, this part right here is what we're talking about today, right? This part right there. And these other parts we'll talk about tomorrow. But this part is what we're going to look at today. So what are some things that we can notice about it? It, oops. Well, it looks like we're starting with glucose. That's our primary reactant. And we're going to make some ATP already, just in the first stage. And we're going to be able to harvest some electrons. You remember that represent energy stored within the bonds in glucose? And they're going to be carried by NADH, one of those electron carrier molecules we talked about last time. Also notice, all right, there's a picture of the mitochondria. This whole process of glycolysis, that's the breaking down of glucose, takes place in the cytosol. Right? So this part of cellular respiration doesn't actually take place in the mitochondria. So let's just start with that as kind of a framework for what's going on. So in glycolysis, kind of like hydrolysis, we're breaking something down. And the thing we're breaking down is glucose, sugar. And actually what we're going to do in glycolysis is break glucose in half. So glucose starts out as a 6-carbon molecule represented by this hexagon. And after glycolysis, we're going to be left over with two halves, each 3 carbons big, and each half we call pyruvate. So in glycolysis, we break glucose down into two molecules of pyruvate that are each 3 carbons. 3 plus 3, 6, perfect. So it is the most ancient form of energy capture, that is glycolysis. It's the starting point for all of cellular respiration, right? So it existed out of all of the different parts of cellular respiration the longest time ago. It's been around for the longest time. But it's very inefficient. Glycolysis by itself only gets 2 ATP out of every glucose molecule, which is not very much. It's leaving a lot of stored energy still left in pyruvate. Okay? And like I mentioned, it's the only part that takes place in the cytosol. Right? Maybe think about that. And tomorrow, that is Wednesday, when we have a synchronous session, I'm going to ask when we talk about this and go over what we did in watching this video, why that makes evolutionary sense that glycolysis would be taking place in the cytosol. So, just as a perspective, life on Earth first evolved without oxygen in its atmosphere. You call it an anaerobic conditions. So energy had to be captured from organic molecules like glucose without using oxygen. So organisms that evolved glycolysis are ancestral to all modern life. The first ones existed long ago when there wasn't any oxygen. That's why all organisms now still use glycolysis. It's very ancient. It's been conserved for a very long time, and that's why glycolysis doesn't require oxygen to happen, unlike the other parts of cellular respiration. So this is kind of an overview of glycolysis. It entails 10 different chemical reactions that convert 6 carbon molecules, glucose, into 3 carbon molecules called pyruvate. See here at the beginning, here at the end. Right? The stuff in between all 10 of those reactions we aren't going to focus on very much. All right? But for those of you who are going to go on and maybe become professionals in health or medicine, maybe when you are in college, but definitely when you're in medical school, in medical biochemistry, you will have to memorize all 10 of those steps. You'll have to memorize all of the enzymes that are used in each of the steps. Each of the steps is enzyme catalyzed. And you'll have to be able to draw each of the intermediate products. But for now, we want to focus on the overview. So in glycolysis, we have to actually put in ATP to get it started, that activation energy. But at the end, we make 4 ATP and 2 NADH. Remember, think of these as high energy electrons, stored energy. And we get pyruvate. So this is just kind of an overview. So I can kind of break it down into 2 phases. The energy investment phase, which is endergonic, we're investing some ATP to move the reaction over that activation energy hump. And then the energy payoff phase, which is exergonic. We can harvest 4 ATP and a little NADH. We can harvest some stored electrons, and we get some pyruvate. So if we're talking about in net, these would be our reactants on the left, and these would be our products on the right. So the first half of glycolysis, and this is a little bit in more detail. Okay, all of these blue words with numbers are enzymes that are catalyzing each step of this chemical reaction. Like I said, for us, you don't need to memorize those. You don't need to memorize these black intermediates. But in the first half, we're priming glucose. We're getting it ready to split by phosphorylating and rearranging its bonds. Okay? So that's kind of the idea from the first half. We're adding energy to glucose to phosphorylate it so we can get it in the correct uh, state to break and to release its energy. So there we go. We're highlighting those blue words. Those are all enzymes that catalyze a specific step in the first part of glycolysis. And then in the second half of glycolysis is the oxidation part. We're removing electrons and donating them to NAD plus to make NADH. We also generate ATP directly. Here if you see, and here if you see. So those are for ATP. All right, like I said, this diagram, again, we have all of the blue that are enzymes that catalyze each step. the name of intermediates, and then we have Lewis drawings here of some of the intermediates on the side. But this is more of what we need to remember, this part. So, overview of glycolysis. We have a priming step where we phosphorylate glucose in the first part. We have a splitting step where we break it into two. And then we have an energy harvesting step where we get our NADH and ATP out. So phosphorylate glucose so we can split, and then once it's split, we are able to harvest ATP and NADH energy out of it before we end up with pyruvate. So if you notice, when we're making ATP here at the end, and I'll go back at the end, the last half of glycolysis, we call that substrate-level phosphorylation. So in the last steps of glycolysis, where did the P, the phosphate, come from to make ATP? All right, so the P, the phosphate, is transferred from one of the intermediates in glycolysis. Is called phosphoenol pyruvate, right? It's just one of the intermediates. Okay, and if we go back here, it's right before we get to pyruvate, right? Right before we get to pyruvate. So a special enzyme takes the phosphate group off of it and attaches it to ADP to make ATP. So when we remove phosphate from one molecule and put it on the next to make ATP, we call that substrate-level phosphorylation because there's an enzyme involved. And in this case, PEP or phosphoenol pyruvate would be the substrate. And in that reaction, the enzyme helps that phosphate group being taken off phosphoenol pyruvate and put onto ADP to make ATP, just like this, right? It holds in its active site ADP, it holds in its active site the phosphoenol pyruvate, and it allows the detachment of phosphate and it to be put on ATP. So that's called substrate level phosphorylation, when an enzyme helps take a phosphate group off of one molecule and to stick it onto ATP. We get it directly from the substrate. So let's look over at all what we call the energy accounting of glycolysis, going from glucose to pyruvate. That's the big story. We make, or sorry, we put in 2 ATP in the first energy investment phase to get over our active energy hump to start the energy payoff phase. We make 4 ATP. So in net, in total profit is 2 ATP. All right. We also, if we go back and look at this picture, make 2 NADH. Okay, so here is at the beginning and here is at the end. So the idea is that's not a lot of energy, all right? But for 1 billion years, this is how life on Earth survived, only harvesting about 3.5% of energy that's stored in glucose. And because of that, organisms that were alive at that time, mainly single cellular prokaryotic organisms, grew very, very slowly and reproduced very, very slowly because there wasn't a lot of energy for them to use. All right. But there's actually a lot more energy stored in glucose. So eventually other pathways had to exist to break down glucose even more, and we'll talk about more of them tomorrow when we continue on this path of cellular respiration. But one thing I wanted to stop and think about is, all right, if NAD plus is accepting the electrons from glycolysis and turning into NADH that carries them, are we eventually going to run out of NAD plus? How is NADH and NAD plus recycled? What is that cycle, right? Without regenerating NAD plus from NADH, our energy production would stop. So another molecule must accept those hydrogens from NADH to turn it back into NAD plus so it can go back and be used again. So how is NADH recycled back into NAD plus? All right, in aerobic respiration, or such as ethanol fermentation, lactic acid fermentation, or in aerobic with oxygen. Okay, there are two different ways that it can be recycled. So, with oxygen present, okay, we can recycle some NADH back in NAD plus by it being utilized in other steps of cellular respiration. Without oxygen, it is used in a process called fermentation, which is how a lot of early organisms made their energy because it only utilizes glycolysis. So in anaerobic fermentation, one example is like in ethanol fermentation in bacteria and yeast. When we go through glycolysis and create pyruvate, that NADH is used to reduce pyruvate to ethanol and carbon dioxide. So ethanol and carbon dioxide are waste end products of fermentation, the process of energy creation, ATP creation, in things like bacteria and yeast. In animals and some fungi, like animals like us, when we are in an anaerobic state, think about exercising very intensely, we don't have enough oxygen coming in to allow cellular respiration to be aerobic cellular respiration, that is, to be the main source of ATP. There's not enough oxygen for the rest of the steps besides glycolysis. So instead, we use the little bit of ATP that we created in glycolysis to power our muscles and things. And then the NADH, those stored electrons, were used to reduce pyruvate to lactic acid. If you exercise, you've probably heard of that before, lactic acid, which is a waste, a byproduct. And then lactic acid is shuttled to our liver and is regenerated into glucose through a process called the Cori cycle. The Cori cycle. So pyruvate is really the branching point. If oxygen is not present, anaerobic conditions like existed on early Earth, fermentation is the end product. We reduce pyruvate to ethanol or lactic acid, all right, and then that's kind of the it. If oxygen is present, we can get much, 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 much more energy out of pyruvate, and we go and end up doing the Krebs cycle and the electron transport chain and all of that, which we will talk about later this week. So we aren't going to say much more about fermentation. We're going to stop with what we know today. And the rest of us talking about cellular respiration has to do with these steps here because there's much more involved than just the end point of fermentation. So hopefully we saw today how we start initially getting ATP energy from glucose through the process of glycolysis.